Hello friends, in this video we are presenting explanations of Jamia Millia Islamia 2021 MSc Bioscience paper. Let's begin. Which of the following has been basic to the origin of life? Carbohydrate, proteins, nucleic acids or nucleoproteins? As we know, nucleic acids and proteins, these are the two basic components to sustain life, especially nucleic acids, because these are the information molecules which are required to build self-replicating entity which is absolutely necessary to continue the generation. Hence, option C would be the correct answer. Which gas was not present in prebiotic environment? Methane, oxygen, carbon dioxide or sulfur dioxide? Urey and Miller in their classical experiment simulated the primitive earth conditions and proposed that biomolecule must have originated from methane, hydrogen and water vapors. Definitely oxygen must not be present in that environment as photosynthesis was not possible. So hence option B would be the correct answer. Diastole is increase of the heart muscle, relaxation of the heart muscles, contraction of the heart muscles or none of these. As we know, diastole is the relaxation of ventricle and pumping of the blood into the heart chambers, while systole, which is the opposite of it, that means it is the contraction of the heart muscle to pump out blood in the entire body. Hence, option B would be the correct answer. Which of the following gland is often referred in relation with AIDS? Thyroid, thymus, adrenal or pancreas? Thymus gland, which plays a key role in the maturation of T cells, is often implicated in AIDS. Remember, AIDS virus, HIV virus, it attacked the T helper cells. So hence option B would be the correct answer. Character of Klinefelter syndrome is, that's very easy. Klinefelter syndrome in which the male baby was born with an extra chromosome. Hence the option D would be the correct answer, that the character of a Klinefelter syndrome is XXY. Vitamin D is synthesized in skin by the action of sunlight on cholesterol, pseudocholesterol, orthophenoxycholesterol or 7-dehydroxycholesterol. Vitamin D3, which is cholecalciferol, is synthesized in skin by the reaction of 7-dehydroxycholesterol with the help of UVB radiation of sunlight. So hence option D would be the correct answer. Hinny is an offspring of male donkey and mare, lion and tiger, tiger and lioness, stallion and female donkey. Whenever a cross between a stallion, that is a male horse, and jenny, which is a female donkey, is being crossed, then the production of hinny takes place. While if you are taking a mare, that is a female horse, and jack, that is a male donkey, then there is a production of mule takes place. So in this case, the hinny is an offspring of a stallion and female donkey. Genetic drift operates only in island population, smaller population, larger population or Mendelian population. So first of all, we should know what is genetic drift. It is a change in allele frequency in a population that happens due to the chance event from one generation to another. It is generally more pronounced in smaller population where it may trigger variants of the gene to fully vanish and hence it minimizes the genetic diversity. That's the reason why genetic drifts operates only in the smaller population. Spindle fibers of mitotic cells are made up of, they are made up of tubulin. So hence the option D would be the correct answer. Thymine dimers are produced due to a DNA damage caused by UV light, X-ray, oxidizing agents or alkylating agents. Generally, pyrimidine dimers, these are the molecular lesions which are formed due to the UV light, generally UVC light, which may cause the formation of covalent linkage between the two adjacent pyrimidines or generally thymine. So hence the option A would be the correct answer. Organisms phenotypically similar but genotypically different are called as multizygous, homozygous, homozygous or heterozygous. Organisms which are phenotypically similar but genotypically different, these are called as heterozygous. While if the organism is phenotypically as well as genotypically similar, then such organisms are homozygous. So here the option D would be the correct answer. Nobel Prize for jumping genes was awarded to Kornberg, McClintock, Khorana or Watson. Jumping gene, which is also a DNA sequence that moves from one location on the genome to another, also known as transposons. The concept of transposons was introduced by Barbara McClintock when she was working on her studies of kernel's color of corn. So where she introduced this concept of transposons. So hence option B would be the correct answer. Which of the following is not a method for horizontal gene transfer? Binary fusion, transformation, conjugation or transduction. First of all, you should know that the gene transfer can be done by two methods. That means vertical gene transfer and horizontal gene transfer. Vertical gene transfer में क्या होता है? Here the organism transfer its gene which is being reflected in its progeny, like father transfer its genes which is being reflected into his son. But in the case of horizontal gene transfer, an organism when it transfers its gene, it is being reflected into the other organism of his own generation. For example, a bacteria can transfer its genes into the another bacteria of his own time, that is called as conjugation. A virus may transfer its gene into another bacteria, that is called transduction. Or a foreign DNA can be transformed into another bacteria called transformation, like we do it in the RDT. Binary fission is not a method of horizontal gene transfer. Hence option A is the correct answer. Degenerate codon is when the codon degenerates soon after synthesis of protein. The codon that code for more than one amino acid. The same amino acid can be coded by more than one codon. A codon is non-functional and is also known as nonsense codon. Genetic codon is degenerate or redundant if a single amino acid can be coded by more than one codon. So hence the option C would be the correct answer. Gene amplification is, it is the production of a new genes within a genome, transfer of other genes to a genome, modification of a gene for desirable purpose, or the production of multiple copies of a gene within a single genome. Gene amplification, it refers to the natural or artificial process by which number of copies of a gene is increased, but without a proportional increase in the other genes. So hence the option D would be the correct answer. The term cDNA library means the pool of cDNA generated from a specific tissue inserted in an appropriate vector, compilation of cDNA sequence in a database, collection of cDNA clones by an individual researcher, or it is a manual for cDNA research. cDNA or complementary DNA library, it is a collection of complementary DNA fragments representing the RNA molecule present in a particular preparation obtained from a given cell or a tissue type. So hence option A would be the correct answer, that the term cDNA library simply means the pool of cDNA generated from a specific tissue inserted in an appropriate vector. Molecular chaperones are a group of proteins that are used in biotechnology for modifying the biochemical reaction, formulation of new proteins, unfolding of proteins, or folding of proteins. Molecular chaperones, these are ubiquitous and highly conserved proteins present in any given cell which facilitate the protein folding. Hence, the option D would be the correct answer. The sugar base of a nucleotide is a pentose, hexose, haptose, or triose. If you see the diagram, the sugar here would be a pentose sugar, which is generally in the case of DNA, it is a deoxyribose, while in the case of RNA, it is a ribose sugar. So hence, the option A would be the correct answer. Molecular scissors used in genetic engineering are helicases, DNA polymerases, restriction enzymes, or DNA ligases. Restriction enzyme, they are also known as molecular scissors. Why? Because they cut at a specific sequence present on a DNA. Generally, such sequences are palindromic sequences. For example, a famous restriction enzyme, that is EcoR1, it recognizes a sequence called GAA-TTC. So hence, restriction enzymes are the molecular scissors. Cell division can be temporarily suspended by radiating with UV light, heat treatment, by depriving cells with CO2, or treatment with colchicine. Colchicine, it is an antimitotic drug where it prevents spindle fiber formations. Some other examples of these such kind of drugs are vinblastine, grisofulvin, etc. So hence, cell division can temporarily be suspended by using colchicine. Housekeeping genes are. So out of 4 attributes, we have to find which one suits better for housekeeping genes. They are constitutively expressed, selectively expressed, randomly expressed, or never expressed. Housekeeping genes are typically constitutive genes that are always expressed because they code for those proteins which are constantly required by the cell. So hence the option A would be the correct answer. Codons causing chain terminations are. Generally, termination codons are UAG, UGA and UAA. Hence option C would be the correct answer. Hybrid vigor is mostly due to heterozygosity, superiority of all genes, homozygosity of pair of characters, or mining of cytoplasm of male with that of a female exclusively. Increase in characters like size, growth rate, fertility, or even yield of a hybrid organism over those of a homozygous condition generally called as pure lines parents resulted from heterosis or hybrid vigor. Generally, such kind of a heterozygous condition is often enjoyed by the plant breeders, where they conditionally make such plant species which are heterozygous in nature and are having better superiority in terms of various characters. So hence option A would be the correct answer. An incomplete dominance was shown in sweet pea, 4 o'clock plant, mustard, or wheat plant. Incomplete dominance is a form of intermediate inheritance in which one allele for a trait is not expressed completely over its paired allele. For example, flower color of a Mirabilis jalapa, also known as 4 o'clock plant, where if you cross a red plant with a white plant, then F1 generation would be the intermediate character, that means pink color. So hence, incomplete dominance, so the best example is Mirabilis jalapa or 4 o'clock plant. Chromosomes having equal arms are known as acrocentric, metacentric, telocentric, or concentric. If you see in the figure, based on the position of centromere, chromosomes can be categorized into various categories like metacentric, where the medial centromere is there and the chromosomes are having equal arms. Submetacentric, where the subterminal centromere causes one short arm and one larger arm of a chromosome. Acrocentric, where the centromere is terminal and one of the arm is very large while another arm is very small. So hence chromosomes having equal arms are known as metacentric or option B. Ethidium bromide acts as a mutagen by substituting adenine by its structural analog? No. Chemical modification of base? No. Production of inter-strand crosslinks in DNA? No. Intercalating between adenine thymine base pair of DNA bases. Basically, ETBR is a mutagen because it intercalates between the DNA and affects the biological processes. Hence, option D would be the correct answer. A competitive inhibitor binds at the active site, does not bind at the active site, alter Vmax only, binds at allosteric sites. Competitive inhibitors are the structure analog of a natural substrate of an enzyme, and generally they compete for the active site and often cause an increase in Km while do not change the Vmax. Hence option A would be the correct answer in this case. Which of the following enzyme increases in serum after heart attack? So we have to find out कौन सा enzyme है generally जिसका level बढ़ जाता है after heart attack. Creatine kinase, SGPT, alkaline phosphatase or amylase. Truly speaking, cardiac marker proteins are troponin T and troponin 1 because hearts only makes these proteins. But at times, doctors also check for the level of creatine phosphokinase or myoglobin in some conditions. Creatine kinase myocardial band, or also known as CKMB, it is a characteristic marker for myocardial infarction, and its level rises after 12 to 24 hours of heart attack. So hence option A would be the correct answer. In fibrous protein, polypeptide chains are folded, twisted, coiled, or extended. Fibrous proteins, they form the skeletal network and generally dominated by the hydrophobic amino acids, and they remain extended just like you see as the here it is. So here is the best example of a fibrous protein, and it is in the extended form. So hence option D would be the correct answer. Glucose absorption in intestine requires a carrier and sodium, a carrier and potassium, sodium only or carrier only. Generally, the glucose absorption in intestine requires a sodium glucose transporter, which is also a co-transporter, which actually transport a glucose uphill. That means from lower concentration to the higher concentration, the energy of this transport is derived by the downhill movement of sodium ion from intestinal lumen to the epithelial cells. Hence, glucose absorption in intestine requires both a carrier and a sodium ion. Most of the amino acids found in human bodies are, that's very simple, they are L isomers. So option A would be the correct answer. Always remember, in natural system, most of the amino acids are in L form, while most of the carbohydrates are in D forms. O-glycosidic bond is formed when a sugar reacts with acid, a sugar reacts with alkali, a sugar and anomeric carbon of sugar reacts with an alcohol, a anomeric carbon of sugar reacts with an acid. O-glycosidic bond कब बनता है? O-glycosidic bond is formed when a hydroxyl group on anomeric carbon of the sugar reacts with an alcohol or an amine of another molecule, where it gets condensed and form an O-glycosidic bond. So here the option C would be the correct answer. The concentration of IgE class of immunoglobulin increase in blood in allergic reactions, diabetes, cancer, or cold condition. Always remember, the IgE class of immunoglobulins, they are associated with allergic reactions or type 1 hypersensitivity. These antibodies, they basically sits over the basophil or mast cells and often cause allergic reactions against allergens like pollen grains, food particles, fungal spores, or pet hairs like denders. So option A would be the correct answer. Lymph is modified blood that contains RBC and WBC, RBC, WBC and proteins, WBC and all proteins, all content of blood except RBC and certain blood proteins. Lymph is basically modified blood and contains all components except RBC and platelets. So hence option D would be the correct answer. Ubiquitin is a protein required for protein degradation, fat degradation, carbohydrate degradation or protein synthesis. Ubiquitin एक 76 रेसिड्यू लॉन्ग पेप्टाइड होता है which whenever it is attached to a substrate जिसको हमें डिग्रेड करना है, so then such substrate is being degraded in the lysosomes because ubiquitin peptide उस पे attached होता है. So hence ubiquitin is a protein required for the protein degradation. Option A would be the correct answer. Connection between arteries and veins is through venules, arterioles, capillaries, or none of the above. Arteries, they generally carry oxygen rich blood away from the heart, while veins, they bring back this deoxygenated blood back to the heart. At tissue level, both arteries and veins, they are connected by the capillary network. So hence the option C would be the correct answer. Ribosomes are attached to the surface of endoplasmic reticulum by covalent action, hydrostatic force, ionic interaction, or through a glycoprotein called ribofourin. Always remember, in eukaryotes, the rough endoplasmic reticulum has ribosomes over its surfaces. Generally, this attachment is facilitated between ribosomes and ER is through a hydrophobic protein, that is signal recognition particle. which is linked to the 60s subunit of the ribosomes, while the ER is connecting with the ribosomes is through the SRP receptor. That means क्या याद रखना है, signal recognition particle will be present over the ribosomes, while इसका receptor जिसको also we call it as ribofourin, it will be present over the ER. Hence the option D would be the correct answer. Phagocytosis involve production of superoxide, H2O2, superoxides and H2O2, or none of these. Lysosomes, they are generally involved in the phagocytose microorganism killing. और ये जो phagocytose microorganism killing है, that can be of two types, oxygen dependent and oxygen independent. When we say oxygen dependent, that means we are going to produce all such species which are going to ultimately produce the oxide radical. For example, superoxide, H2O2, hydroxide radicals, hypochlorite, nitric oxide और other antimicrobial compounds, और वो एक तरह से रेस्पिरेटरी बर्स्ट होता है. While in oxygen independent killing में क्या होगा, there would be no production of superoxide and H2O2. Rather, we will be using the hydrolase enzyme. So phagocytosis involve production of superoxide oxides and H2O2, but in the oxygen dependent the killing. So hence option C would be the correct answer. Ciliated epithelium is found in trachea, urinary bladder, intestine or stomach. Ciliated epithelium is found in the respiratory tract and fallopian tubes of the female, which helps in the movement of particles or eggs. So hence option A would be the correct answer, that in the trachea ciliated epithelium can be found. Other than that, the fallopian tube is the option. In metalloenzymes, metals are attached to the enzyme through coordinate bond, attached to the enzyme by covalent bond, attached to the enzyme by non-covalent bond, loosely attached to the enzyme. In metalloenzyme, the metal complexes are attached to the protein core through a coordinate bond, where one of the metal ion is having one labile coordination site. So hence option A would be the correct answer. An example of N3 fatty acids are palmitic acid, arachidonic acid, linolenic acid or linoleic acid. Omega-3 fatty acid or also known as N3 fatty acids, they are the important dietary component हमेशा जरूरत होती है present in certain food. Some of the example of omega-3 or N3 fatty acids are alpha linolenic acid, that is ALA, icosapentaenoic acid, that is EPA, or docosahexaenoic acid, that is DHA. So here the option C would be definitely the correct answer. ALA or alpha linolenic acid is also is C18, where the 3 double bonds are present with one terminal double bond 3 carbons away from the end. So hence the option C would be the correct answer. Always remember 3 omega fatty acid होते हैं, generally alpha linolenic acid, icosapentaenoic acid and docosahexaenoic acid. You have to remember that. Polypeptide bond formation occurs during starvation, from amino terminus, from carboxy terminus, from amino terminus to carboxy terminus. Peptide bond is formed between COOH group and NH2 group of another amino acid. Hence, every time whenever a peptide bond formation occurs, it will be having one NH2 group at the one end and COOH group at the other end, which signals the end of the peptide synthesis. So, हमेशा polypeptide bond formation कैसे होगा? From one amino terminus to the carboxy terminus. Always remember that. So, option D would be the correct answer. For an exothermic reaction, the increase in temperature will shift the equilibrium towards low volume side, high volume side, temperature has no effect, or towards product side. See, for an exothermic reaction, heat is the product of reaction. तो अगर हम increase करेंगे temperature of such reaction, तो as per Le-Chatelier principle, reaction will proceed towards the backward direction. Now, it appears to us that no option is correct here. But if we think, let's say, since reaction are moving in favor of the reactant which are more ordered, so obviously if they are more ordered, they will occupy the less space or less volume. So we can say that reaction will shift in favor of low volume side. So hence option A could be the correct answer. But remember, this answer can only be valid if we will be forced to find out the answer from these only. Which is most acidic? Phenol, benzyl alcohol, cyclohexanol, or metachlorophenol? That's very easy. Metachlorophenol will be the most acidic example. Why? Because electron withdrawing nature of a chlorine, it will increase the acidic character of a metachlorophenol in comparison to phenol. So hence the option D would be the correct answer. The bond that determines the secondary structure of a protein is coordinate bond, hydrogen bond, covalent bond, or ionic bond. Always remember the primary structure of a protein. It is being determined by the covalent peptide bond. But if we have to make the secondary structure, then we have to provide the hydrogen bonding between the carbonyl and the hydrogen attached to the nitrogen of a peptide bond. So hence the hydrogen bond between the peptide backbone is the reason why secondary structure forms. Remember, there is no role of a side chain in the secondary structure formation. They play an important role in the tertiary structure formation, but not in the secondary structure formation. So hence the hydrogen bond would be the correct answer here. If U-235 is bombarded with neutron, the atom can split into strontium and lead, cadmium and krypton, cesium and rubidium, barium and krypton. So if we see the radioactive decay of U-235 with a neutron, you'll find that the initially U-236 species will be formed, which is apparently degraded into the barium and krypton. So hence option D would be the correct answer. Hydrolytic reaction of fats with caustic soda is known as saponification, esterification, carboxylation and acetylation. एक और इजी क्वेश्चन है, अगर आप जामिया के पेपर को पूरा देखें तो आप देखेंगे कि बहुत सारे इजी क्वेश्चंस हैं which you can easily attempt. Here the answer would be the saponification. Saponification is a process by which the vegetable oils or animal fats is reacted with strong alkali to form the soap, which you always read in the lower classes. So hence the option A would be the correct answer. Which of the following aqueous solution has highest boiling point? 0.1 molar KCl, 0.1 molar barium chloride, 0.1 molar aluminum chloride और 0.1 molar Na2S. एक और इजी क्वेश्चन केमिस्ट्री से पूछा गया है. As you know, the elevation in boiling point is a colligative property of the solution. That means जब भी boiling point elevate होगा, it will be directly proportional to the solute particle concentration. अब अगर हम नॉन वोलेटाइल सब्सटेंस को डिसॉल्व करेंगे किसी सॉल्वेंट में, तो बॉइलिंग पॉइंट ऑफ द सॉल्वेंट इज बाउंड टू इंक्रीज. Higher the concentration of a particle, there will be that much amount of increment in the boiling point would be there. अब आप देख लीजिए किसके अंदर पार्टिकल कंसंट्रेशन ज्यादा है. If you see, in case of 0.1 molar aluminum chloride, number of particles will be highest. Hence, the option C would be the correct answer. For an ideal gas, delta H by delta P at constant temperature is, always remember, for an ideal gas, the value of delta U by delta P at constant temperature is equal to the delta H by delta P at constant temperature, and the value is always equal to the 0. So hence option A would be the correct answer. The necessary condition under which a reaction is at equilibrium, that's very easy. That is always be equal to the 0. That means delta H at constant temperature and pressure should be at 0 if the reaction is poised at equilibrium. So hence option A would be the correct answer. Aspirin is phenyl salicylate, phthalic acid, salicylic acid or acetyl salicylic acid. Always remember that aspirin is also known as acetyl salicylic acid. So hence option D would be the correct answer. The following are positively charged amino acids except aspartate, arginine, lysine or histidine. That's very easy. Histidine, arginine and lysine, these are the positively charged basic amino acids, while aspartate is the negatively charged amino acid. Hence option A would be the correct answer. An example of sedimentary type of nutrient cycle is nitrogen cycle, phosphorus cycle, carbon cycle or none of these. Do you know what is sedimentary cycle? These are those cycles in which the main reservoir of chemicals are soils and rocks, which includes minerals, for example phosphorus and sulfur, which is the best example of sedimentary type of nutrient cycle. Lignin is a component of secondary cell wall of epidermis, collenchyma, sclerenchyma or parenchyma. Lignin is the component of secondary cell wall component of sclerenchyma. The cell wall of sclerenchyma contains cellulose, hemicellulose and lignin. So hence option C would be the correct answer. Phytoplanktons are producers of forest, producers of lake, producers of desert or consumer of sea ecosystems. Phytoplanktons, these are the photosynthesizing microorganisms found in the upper layer of freshwater bodies, hence also called as producers of freshwater bodies. So option B would be the correct answer. Which will not cause air pollution? Sulfur dioxide, carbon monoxide and nitrogen. ये तो बहुत इजी है. नाइट्रोजन हैज नो पॉल्यूटिंग इफेक्ट. इनफैक्ट हमारे एटमॉस्फेयर में नाइट्रोजन सबसे ज्यादा होता है. तो अगर इसने एयर पॉल्यूशन करना होता तो आज हम जिंदा नहीं होते. तो हेंस ऑप्शन डी वुड बी द करेक्ट आंसर. Minamata disease was first reported from Japan, USA, China or Canada. Minamata disease was first discovered in the city of Minamata, Japan in 1956. It was caused due to the release of methyl mercury in industrial wastewater from a chemical factory. Hence, option A would be the correct answer. Blue baby syndrome is caused due to the excess of nitrogen, copper, iron, phosphorus. Blue baby syndrome, which is also known as methemoglobin, it is caused by drinking water which is contaminated with nitrates. Since nitrate is not given, so we can assume that nitrogen would be the correct answer. So option A is the correct answer here. Cadmium poisoning cause Blackfoot disease, goiter, SCOBY or itai. Cadmium poison, it caused itai syndrome, which was first reported in Japan. The person who was having this disease, they have severe joint pain and they generally refer it ouch disease or itai disease. So hence option D would be the correct answer. The example of nitrogen fixing cyanobacteria is Pseudomonas, Anabaena, Rhizobium or Clostridium. If you see in the diagram, all these are example of cyanobacteria which can fix the nitrogen. Here you can find the Anabaena. So hence Anabaena is an example of nitrogen fixing cyanobacteria. With this, we conclude first part of this series. Stay tuned and subscribe for more updates. If you have anything specific, then do write to us. Thank you very much.